Hi, we're going to be talking today about sampling for chlorophyll in seawater. So of course, we're sampling for chlorophyll in this activity. What we're really doing is trying to quantify or get a relative abundance of the amount of phytoplankton in the water. So in class, we talked about the importance of phytoplankton, where we find it throughout the oceans, and in this module we're going to be talking even more about the factors that control primary productivity and how productive phytoplankton are as the base of the food web. Right now, the lab is about how we actually go out and quantify how much phytoplankton are in the water, and we do that by quantifying the amount of chlorophyll that we find in the seawater. Phytoplankton have to have chlorophyll to do photosynthesis, so it's a great proxy for the relative abundance of phytoplankton algae in the water. So here's a sample that Carl collected from Mission Bay. Now, very often, water samples we use in marine ecology are relatively small, something like in this size, like about a liter. When we sample for chlorophyll, especially in the wintertime, when the sunlight is lower, we often get samples that are much larger, like this, about 4 liters in size, about a gallon or so. Because in order to get enough chlorophyll to quantify, we need to filter a large volume of water. This was collected off the side of a boat. We usually, in this lab, collect these water samples in shallow water at the surface and then deeper down at the bottom, and then compare the amounts of phytoplankton between those two depths. So we're going to show you how we go ahead and process this sample to quantify how much chlorophyll is in it. So we're going to go over here to our filtering apparatus. The first job is going to be to take this water sample and filter it down to try to collect all the phytoplankton that are in this sample on a filter that we can then process for chlorophyll. So here we have our filters. These are Wattman glass microfiber filters. So they're basically small discs of glass fiber. They look like paper, but they're actually glass fibers. And they're very, very sort of porous, and they trap very small particles down to several microns as the water is able to flow through these filters. And so the way we use this is we take our glass fiber filter. We're going to pick it up using tweezers so we don't get our oil from our hands on this. And we're going to go over here to our filtering apparatus, put this down, and then we're going to attach our glass cup and lock that down on there like that. Now we have something we can pour the water into on top of the filter. The water can go down through the filter, leaving us with our phytoplankton. Okay, so when we filter a sample, we need some suction. We need some vacuum. The water will eventually drip through this filter, but it'll take forever unless we actually motivate it somehow with some vacuum. So this is our suction or filtering apparatus. We have the ability to filter about 6 samples at a time. Today we're only going to do 1. And connected to this cup where we pour our water are these tubes, eventually connected to a vacuum supply over here. So I'm going to actually turn that on. And now our vacuum is running here, so the air is being sucked through the filter and down in this direction. I'm going to put a little deionized water in there to start this going. And then we're simply going to take our water sample and we're going to pour this into this cup, a little bit at a time, and then let it be sucked down through the filter. And if you're zoomed in enough, you can actually see the water is slowly going down as it travels through the filter. So we've sucked our sample down. This is the last little bit of it. 4 liters of water. And after this filters all the way down, we'll have our sample ready on our filter. Okay, so here's our finished filtered seawater. You can see the green and brown on the filter representing lots of algae, phytoplankton, as well as maybe some sediment. And these can be stored until they're ready to be processed for up to a month. So we do is we put these in aluminum foil, we fold them over, and then we seal them up like this, put a label on the aluminum foil so we make sure we know where this is collected from and when, and then store these for up to a month in the freezer until they're ready to be processed. So here's our filter once again, and what I'm doing is cutting away the outer part of the filter that doesn't have any sample on it. What we're going to try to do with this is we're going to try to dissolve away the glass fiber part of this and retain just the chlorophyll that's on here. And so to do that, we're going to actually use acetone as our solvent. So the first step is to try to cut this into tiny little bits so that it's as easily to dissolve as possible. So our first step is to take this filter and just use your scissors to cut this into very, very small pieces that go into this tube. So here's our filter, all nice and cut up into small bits. In this tube, and I'll put some acetone in the tube, which is going to help start to dissolve away the paper and retain the chlorophyll. We're going to want to pulverize this or mash this up so it's even extracting even more effectively to do this. So I'm going to go over to this grinder. We're going to turn this on at a relatively slow speed. And all we do is put their tube into here and let this mash up these little bits of paper for a few minutes. So by moving the tube around and the walls of the tube against this grinder, we can basically pulverize the paper and better expose all of the phytoplankton cells to the acetone. All right, so we've been grinding this sample now for a few minutes. It's pretty well pulverized. And what I'm going to do is I'm going to stop the grinder. I'm going to pull this out, use a little bit of acetone to carefully just get any little bits that might be on the grinder down into the tube. And then we're going to decant this into a centrifuge tube. So we're just going to pour off everything that's in here, the liquid and the little bits that are solid that are still left, into the centrifuge tube. And then the centrifuge tube can be stored also frozen in the dark for a period of time before we process this. In fact, it's actually one thing we have to do, is keep this in the dark, usually in the freezer, for at least 24 hours before we process the sample. That gives the acetone a chance to dissolve away lots of the paper, extract the chlorophyll, and it's going to give us a better result when we go to use this. Okay, next step is going to be to centrifuge our samples. So here are a number of chlorophyll samples that have been sitting in the freezer, extracting in acetone, right? And we have them labeled so we know where they came from. And we're simply going to put these in our centrifuge, and we're going to spin this so that our liquid goes down or is retained, and all the solids go down to the bottom of the tube. So the solids will go to the bottom of the tube, the liquid will remain, and we can then use our spectrophotometer to analyze the liquid, the color in the liquid. The most important thing about using a centrifuge is to make sure it's balanced. So we've got 4 samples here in our tubes, and they're across from each other, so they're balanced. And just for safety's sake, I'm actually going to fill up every chamber here and make sure this is completely balanced, so that when it spins, there's no issues with it wobbling. So there's our samples in there. We're going to turn our centrifuge on. Down here, I can show you that it's spinning right now. We spin this for 2 to 3 minutes. That's plenty of time for the solids to be cast down to the bottom of these tubes, and then you can then use the liquid for our analysis. Okay, centrifuge is done spinning. Got our samples here. And here's the sample we've been working with. You can see all the solid material down at the bottom of the tube, and all the green liquid containing chlorophyll at the top of the tube, the supernatant. And our goal was to try to be able to use the supernatant in our spectrophotometer to read the absorption of wavelengths of light in this sample. That's why we had to centrifuge this to get all the material down to the bottom. So let's bring this on over to our spectrophotometer. So spectrophotometer is a device that lets us read either absorption or transmittance of light through a sample. And our first step is going to be to zero the spectrophotometer. And because we extracted this in acetone, what we're going to do is use cuvette filled with acetone as a blank. So our first step is to put this in here. We're going to close our cover. We've already set the wavelength to the first wavelength of what we want to read, which is 480 nanometers. And we're going to hit the 0 button. And this is going to zero the spectrophotometer. In other words, it's going to say that 0 absorption is a blank full of acetone. We can compare that to our sample containing our seawater and chlorophyll. Acetone containing chlorophyll. Here's our centrifuge sample. I'm handling the cuvette at the top so I don't get my grease from an oil from my fingers on it. We're going to carefully decant this. We need the bottom to be filled. And there's our sample we're going to put in our spectrophotometer. We just zeroed the spectrophotometer. We're going to pull that out. We're going to put the sample in, close the lid, and we're going to allow the spectrophotometer to read the absorption. Again, what we're reading is how much light at this wavelength, 480 nanometers, is absorbed by our sample. So our reading right now is about 0.38. So we're going to record that number. And then we're going to go ahead and we're going to read the absorption at other wavelengths that are specified in your lab handout. And we're going to use those readings in an equation to figure out how much chlorophyll is in our sample. All right, so we have a sample in there right now. We're at 480 nanometers. And one thing on this display screen you can see is the color spectrum at the bottom. So remember, the wavelengths of light represent different colors in the visible light spectrum. What we're doing is looking at the absorption at different colors of light or wavelengths of light. So remember that chlorophyll and plants and algae that use it look green because that green light is reflected to our eyes. So the maximum absorption is going to be in blue and yellow and red wavelengths. And the maximum reflection is going to be at green wavelengths. So if we go ahead and change the settings here up into green, we should see things change. All right, so now there's very little absorption at those wavelengths because that light is reflected. And if we go even farther up into the higher wavelengths of light, into yellow, that represent yellow, orange, and red, we should see an increase in the amount of absorption. Okay, so we have our first reading of 480 nanometers. We're going to pull this sample out, and we're going to put in our acetone blank once again. We're going to turn our wavelength up to 510 nanometers, which is our next reading. Okay, 510 nanometers. And I'm going to re-zero. So I'm going to press the 0 button once again and get the machine to recognize acetone at this wavelength, using this wavelength as 0 absorption. Once it's ready, and it is, we're going to proceed the same way we did before. We're going to pull this out, handling this only at the top of the cuvette. We're going to put this in, let our machine read absorption, and record that number. And then, as you see from your handout, we're going to repeat that procedure for several other wavelengths and get an absorption reading in all of those wavelengths.
All right, everybody, that wraps up Lab 3 chlorophyll analysis. Thanks for your time, and I'll talk to you all soon.